Dr. David Weller is the Research Director of the USDA Agriculture Research Service Biological Control and Root Disease Research Unit at Washington State University in Pullman. He's a fellow of the American Phytopathological Society and of the American Academy for the Advancement of Science. He's published over 150 papers and review articles and is well known for his STEM outreach. He has been involved with our CommGen Authentic Research Experiences grant. His lab analyzes our data and does further research on it. We are very, very excited to have him here speaking with you today.
I know you have heard from Dr. Bengera about the basics of take-all decline and the process of suppression of take-all disease in wheat. And today I'd like to introduce you to some of the topics of basically what's new in this study of natural suppression of take-all disease of wheat. And then after myself, Dr. Tomashow will be talking about phenazine producers in Pseudomonas phenazine producers in dryland wheat. But I just want to first give you the update on what's new in the exciting world of take-all decline. I like to always begin my presentations to students and scientists around the world with this slide. And it really shows the diversity of people around the world. But the point here is that we know that by the year 2050, there will be about 9 to 11 billion people on Earth to feed using the same amounts of, or less of land and water that is now available for agricultural production. So there's going to be, in the next 30 to 40 years, a lot more people on Earth that we're going to have to feed. And it is the job of us in science, in the microbiological sciences, agricultural sciences, plant and animal sciences, and veterinary sciences, to make sure that we are able to feed the growing population here on Earth. I think it's really important when we think about the future population in agricultural production that we remember that even today, 1/3 of all agricultural commodities that could be produced are lost to diseases, weeds, insects, and other pests. And soil-borne pathogens and soil-borne diseases, which take-all is a most important one, the most important root disease of wheat worldwide. Soil-borne diseases are cause root and crown rots and wilts and damping off, account for a significant amount of the losses of food, fiber, ornamental, and biofuel crops. And we are going to have a world in the next 20 to 30, 40 years where farmers are going to be challenged to increase production for the growing population, but to do so with less pesticides, less fumigants, and many more sustainable practices such as no-till, precision farming, and biological control. Now, as you have heard many times from Dr. Bengera and your instructors, the topic of the CommGen project is one about wheat. And wheat is one of the most important grain crops grown worldwide and here in the United States. In the state of Washington, which is a major wheat growing area in the country, wheat is grown in all of our agroclimatic zones, but we're going to center our attention on the east side of the state, more in the area as you go over the Cascade Mountains and then go to the Idaho border. I just wanted to refresh with you the fact that in Washington state, wheat is a cash crop worth now well over $1 billion. And it is grown under irrigation, under high rainfall, and under low rainfall in dryland areas. Now, along with wheat, an important part of the topic of the CommGen research project is take-all. And you've learned a lot about take-all in the past, and take-all is caused by Guaminomyces graminis vartritici. And as I have said, it's the most important root disease of wheat, not only in the U.S., but worldwide. And you have also learned from the past that the role of strains that produce 2,4-diacetylfluoroglucanol, or DAPG, are involved in this natural suppression of take-all that we call take-all decline. Now, Dr. Tomashow, later in this series, will talk to you about phenazine-producing strains of Pseudomonas, but I'm going to focus only on the fluoroglucanol or DAPG producers in telling you what is new in this research. But before we get into that, I'll just again refresh your mind on this tremendously exciting phenomenon known as take-all decline. And take-all decline is that phenomenon where disease increases with monoculture of wheat or barley and then spontaneously declines. And you will see that as long as wheat is grown in monoculture, the disease will remain suppressed due to the buildup of 2,4-diacetylfluoroglucanol producing strains. You have probably seen this slide in the past, and it shows the tremendous diversity that occurs with DAPG producing strains in agriculture worldwide. And I wanted to again reiterate that it is in fact the D genotypes that are responsible for take-all decline in the state of Washington and the Pacific Northwest. Now, one of the new things that has come out from the CommGen research done by students at the various colleges that are participating in this project has been the discovery that the strain responsible for take-all decline, which we used to call Pseudomonas fluorescens, but we now call, and it was the D genotype, we found that the strain L51-96 that you're all working on is in fact not a fluorescens, but we can now call it Pseudomonas brassicacearum. And that was one of the first very significant findings that came out from research of students just like yourself. And so I wanted to reinforce the fact of what I have said down here, right? That CommGen research confirmed this very important finding, that the L51 is in fact a Pseudomonas brassicacearum. So thanks for that good work. Now, in the short time I have, I really want to address two new questions that my research team with the U.S. Department of Agriculture, Agricultural Research Service at Washington State continue to work on and have worked on since over the last few years. And there are two common questions that came up. And the first is, I have been asked all over the world the question, if the take-all decline occurs because of the buildup of antibiotic-producing Pseudomonas strains, especially Pseudomonas brassicacearum, and the antibiotic DAPG is produced in the rhizosphere, why then, after years and years of growing wheat and years of monoculture, why does the pathogen, Guaminomyces graminis vartritici, why does it not develop resistance to take-all and to DAPG? Why doesn't it develop resistance to DAPG? And that has been asked over and over. Well, we looked at this question in depth, one of my graduate students, and in that to look at that question, he collected isolates from around the Pacific Northwest, and he collected them from soils that were either suppressive to the disease, they had gone into take-all decline, or from fields that were conducive to take-all. The field had not yet gone into decline. And then after isolating those strains of the take-all pathogen, he tested them on media containing different concentrations of DAPG. And in this slide, we see where we have 0, 1, 2, on up to 5 micrograms per mil. And with this assay, he was able to determine that if you look at isolates, either from the suppressive, marked with an S, the bars with an S, or conducive soils, that you do not find differences among those strains of the take-all pathogen. You do not find differences between those fields, meaning that those isolates coming from suppressive and from conducive soils were equally sensitive to the antibiotic. And this was a bit surprised because we all know in human health how easy it is for human pathogens to develop resistance to antibiotics like streptomycin, penicillin, erythromycin, and many, many different antibiotics that we use to treat our own infections. So he looked at this question more and used a really innovative technique which we call a deletion library of Saccharomyces. And this approach allowed us to ask questions about the mechanism by which DAPG was inhibiting fungi. And what he found after an extensive study was that DAPG is able to disrupt many basic cellular processes in cells. It disrupts cell membrane function, reactive oxygen regulation, and cell homeostasis. And because of this, because DAPG affects so many basic functions in the cell, we realized it would be very difficult for the take-all pathogen to, in fact, develop resistance to this antibiotic. And this is different than many other antibiotics where there may be just one single site of activity. And in that case, it's much easier for resistance to develop. Now, the second question that we have been working on over the last couple of years that has been very important part of our research program with the USDA at Pullman, Washington, is why does the robustness of take-all suppressiveness differ among fields and years? Why is take-all suppressiveness more robust in one field than across the road or in 1 year compared to the next? And we address this question because as I talk to growers and farmers across the country and in fact worldwide, where take-all decline is used as a practical approach to control this disease, this question has come up. Why does the robustness of this suppressiveness differ? And so one of the very interesting possibilities was that the robustness of suppressiveness differs because wheat cultivars differ in their inherent ability to make use of this take-all decline suppressiveness. You may not realize, but in the Pacific Northwest alone, there are over 100 wheat cultivars grown. That's a lot of wheat cultivars grown in just this area of Oregon, Washington, and Idaho. And you can imagine worldwide, there are thousands of different varieties that are currently grown and have been grown over the decades. And so it makes sense that with such genetic diversity in our wheats that are grown for production that you would find differences in the ability to use take-all suppressiveness. And this is exactly what we have found, and I think this is demonstrated very nicely. In this study, we compared the DAPG production on two Washington cultivars, Terra and Buchanan. And what we did, we took these cultivars and we colonized them with Pseudomonas brassicacearum, strain Q8R1-96. Now remember, brassicacearum is what we used to call the D genotype. And Q8R1-96 is very, very similar to your strain L51-96, which you are using in your sequencing project. And so Q8R1 and L51 were not isolated from the same field, but they were both isolated from central Washington from take-all decline fields. But the point I want to really point out here is if you look at Terra and Buchanan, and you look here in the far right-hand side of your screen, you see if you quantify the amount of antibiotic that is being produced on these roots, you will see that Buchanan produces almost or supports almost 6 times the amount of antibiotic produced by Q8R1 than on Terra. So just that fact shows you, even though these wheats are all wheat, there's genetic variability in the generic variation in the ability of these wheats to be colonized by the P. brassicacearum and also to produce the antibiotic. However, one might say, oh, more is better. But more is not always better when it comes to these powerful antibiotics produced in the soil. Just as it is in human medicine, there's a certain dose that is important to have if you're trying to cure pneumonia or strep throat or an ear infection, but you give too big of a dose and it can have some adverse effects. So one of the other exciting work that we're doing to understand why the suppressiveness of take-all decline differs from field to field and year to year is to look at the effect of DAPG, the antibiotic, on the wheat. And Dr. Bengera in her lectures have told you that it's a powerful antibiotic that has activity against bacteria, fungi, nematodes, but DAPG also has a phytotoxic effect. And if you grow wheat on a high enough concentration, wheat will develop these lesions very quickly. And so it can be quite phytotoxic. So there is a balance that is needed in take-all suppression. You have to have a balance where there is sufficient amount of antibiotic produced to inhibit the take-all fungus, but not too much to be detrimental to the plant. It's kind of like the Goldilocks zone. Just the right amount to do the right job. And part of our project now is finding those wheats that tolerate the antibiotic the best, and those wheats are those that can make best use of take-all decline. So I want to finish up by showing you just some more recent data. And this was done by one of my graduate students, Mingming Yang, who's since returned to assume in China a assistant professor position at an important university. But in this study, he looked at the wheat cultivars Terra, Finley, and Buchanan. And in this study, he took either the strain Q8R1 or your strain that you're working with, L51, and he coated the seeds. and he planted them to see the response of those cultivars. And I guess I was really surprised because you will see, if you look on the x-axis, under 10 to the 6th colony forming units of either Q8R1 or L51, you will see that Terra is very resistant to the activity and the phytotoxicity of the DAPG antibiotic. On the other hand, Buchanan is very sensitive to it. And this is measured by the amount of germination. So the bacteria producing the antibiotic while on the seed can have a very detrimental effect on the growth of the wheat. Terra is a superstar. It seems to tolerate the antibiotic produced by Q8R1 or L51-96 very well, and Buchanan's much, much less able to sustain that effect or the damage from the DAPG producer. What we're doing now is looking at all of the wheat varieties grown in the Pacific Northwest to determine which of those varieties are able to best handle colonization by Pseudomonas brassicacearum, which is responsible for take-all decline here in the Pacific Northwest. So growers can make a choice to choose a variety that can make best use of take-all decline, suppressiveness in their field, and wheat breeders can breed this trait into future varieties. So we're really excited that this fundamental work on DAPG and the effect on wheat can be translated into real-world application for farmers. Now, I also want to talk to you about some other exciting work that has been going on. I work at Pullman for the USDA, but I also have an appointment at Utrecht University in Holland, where I have visited many times as an endowed professor. And when I'm there, I'm able to get out of my office and I go back to the lab and I go back to the greenhouse and do my own work. I get my hands dirty again. I use pipettes. I make gels, just like all of you are doing in the lab. And I have to say, after being a professional scientist for 36 years, I never, ever lose the excitement of being in the lab, working in the greenhouse, and doing cutting-edge, real-world science. And that's why I'm so excited about the CommGen project, because it gives you students that opportunity to feel what it's like to work on an important project. And I might add at this particular time that all the work that you do, all those sequences that you generate come back to our lab at Pullman, and we incorporate them into our research project. That's why you were able to show that strain L51 was not P. fluorescens, but in fact was Pseudomonas brassicacearum. So really keep up the good work. We really need the boots on the ground like you of doing this work to keep agricultural research moving forward. But while I'm in Utrecht, I have the opportunity to work on a really exciting system, and that is called biologically induced systemic resistance. And with biologically induced systemic resistance, when beneficial microorganisms colonize the roots of any plant, they are able to induce signals that go up the plant and prime the leaves and the rest of the foliage to be ready for attack. So those roots that have been colonized by certain beneficial bacteria are then able to respond with their defenses much quicker against attack by pathogenic bacteria, insects, as well as fungi. And so, in a way, the plant has an immune system, not like ours, but something very particular to plants. And I'm always excited to be able to work on this. And I have taken the work on take-all decline and moved that work into these questions of induced resistance. So one of the things I do there, I shift away completely from wheat, and I work with Arabidopsis. And Arabidopsis is the lab rat, so to speak, of the plant world. Because of its very small genome, it's very easy to manipulate genetically, and it is a type of plant that's used experimentally throughout the world. So no wheat while I'm in Utrecht, and I work on Arabidopsis, and it's a very different system that I work with. So you take small seedlings of Arabidopsis, you plant them into very small pots in the potting mix, and then they begin to grow, as you see here, large containers of them. And then you can challenge the foliage by dipping the foliage into a bacterial pathogen, which is Pseudomonas syringae tomato. And perhaps it's not as clear here, but you can see on these green leaves, these yellow chlorotic and necrotic spots, and then you can count the number of leaves that have this type of infection. So in this type of a system, I can take your strain, L51-96, or other DAPG producing strains, I can add them into the soil, which then induces resistance against this foliar pathogen in this very beautiful model system. And I can tell you that your strain L51 and other DAPG producers are able to induce resistance very, very nice. So that's a new component to the work that has come out of our work on take-all decline, that these strains not only suppress root diseases, but they also can induce resistance against foliar pathogens. And I just want to share some work that was published a couple of years ago. And in this slide, along the y-axis, you see number or percent diseased leaves. And at the bottom, you have treatments, various bacterial treatments. You'll recognize the treatment of Q8R1-96. And also, you can see, compared to the control on the far left side, it induces real good resistance against the pathogen Pseudomonas syringae tomato. But if you take a mutant that is deficient in the antibiotic, it no longer is able to induce resistance. So this beautiful antibiotic, DAPG, that you're all working with, that beautiful antibiotic has the ability not only to directly inhibit the take-all pathogen, but also it has the ability to induce those very sophisticated mechanisms of systemic defense in plants. And that has been a really major find. And I've been proud of the work because I think it adds a new dimension to what DAPG producers can do. Now, I don't show it here, but your strain L51 was tested in the exact same way and was as good as Q8R1. So it was a really nice study and with really nice effects. Now, I want to end up with this last slide, which is really hot off the press. And indeed, it was hot off the press. Last summer in 2013, I spent another 6 months at Utrecht University as the WCS Endowed Professor of Phytopathology. And following up on my previous work, and what I wanted to do was, in fact, not just use the DAPG producers, but in fact to use that special take-all decline soil from Quincy, Washington. So we took some of that soil over and did the same experiments of into the potting mix with the Arabidopsis. We added in either 20% or 10% of the Quincy take-all decline. So it was 90% or 80% of the potting mix and 10 or 20% of the quincy take-all decline. We demonstrated that the DAPG producers were present, that P. brassicacearum was present, and then we did the assays as we had before. And so we were able to demonstrate for the very, very first time that the take-all decline soil from Quincy, Washington was able to induce resistance in Arabidopsis to Pseudomonas syringae, pathovar tomato. However, when we pasteurized that soil, the two columns on the far right, and we eliminated the DAPG producers, we eliminated that ability to induce resistance against this foliar pathogen. So this work even demonstrates, provides further evidence that in fact, there's more mystery to these suppressive soils, these take-all decline soils, that they're having not only effects on the take-all pathogen, but other pathogens that might attack above. So one thing about science is it's like an onion. You peel back one layer and you think you have the answers, and then you find another question in another question, and that's what science is about. And over the last couple of 3 decades, we've been peeling back the question on take-all decline, and then we find we have even more questions and exciting information that has come about. I want to end up, you may have seen other pictures of my research unit at Pullman, the root disease and biocontrol research unit. In any lab you have, it's like a train moving along. Some of us get on this train and stay for 35 or 6 years. Others come as students and stay for a few years, and others stay a little bit longer. But my point is that we are in a very important time in science and in agriculture. And the excitement that I have about this project is seeing the next generation of scientists, whether you're going into agriculture or plant science, animal science, human medicine, it doesn't matter. The world in this country is relying on you as young people to take up the baton of science, engineering, and technology to move this country forward, to help us feed the world. And I firmly believe that the strength of a country really relies heavily on the strength of its science, engineering, technology, and mathematics. And so that's why I'm pleased to be part of this program. I'm pleased to give you an update. And I hope at some time I will see some of you in my lab working on an internship or maybe being one of our graduate students. So thank you very much for your attention. And I look forward to talking to you in the future as new exciting work comes along and results from our work on take-all decline, DAPG producers, wheat, and we see more of your contributions coming into the lab. So goodbye.